thank you all very much for joining us as usual. As you know, as Eli said, what we try and do in the next hour is have a conversation with someone who's a sort of leading researcher. I'm not so sure I like this distinction between basic and clinical because it seems to imply some sort of hierarchy. But I think the fact that we can all work together to try and better help people with Parkinson's is what the WPC is all around, to bring all the stakeholders together. And we're very privileged today to have Andrew Siderowf with us from Pennsylvania. We're going to have a nice little chat about his life, his work, and how it's changed over the years. And I also want to know particularly about the PPMI, which pops up everywhere, and not everyone may be that familiar with it, but I think you're very heavily involved with that. And I would just like to encourage people to put questions in the chat box, and we will try and get to them as we go along. So, Andrew, you're very welcome. I'm always very interested to know people's background. So I'm always very interested to know, um, where you grew up and how you got into medicine, and then particularly neurology and then Parkinson's. So how
Yeah,
did
so
it begin for you?
thanks Roger, and thanks Eli. It's really nice to be here with WPC, and I was just thrilled to be able to come and join. So I just want to start by saying thank you and thanks to the organization for having me. So how did I get into medicine?
Yeah,
So it's a funny story. So um, I didn't really have anybody in my family who's a doctor. There are a couple of doctors, but not many. My father's actually a champion golfer,
really?
and yeah, he won the
Has he
British
won
Amateur
has he ever
twice.
won the Masters?
No, but when I was a kid, he played the Masters, and we used to go. I
Really?
used to go. We would go and like hide in the bag room, like this, hide behind Jack Nicklaus's golf clubs in the bag room when I was like 8 years old.
Yeah.
Um, and but I wasn't. I knew I couldn't do that. It wasn't that I didn't want to do that, but I didn't have the aptitude for it. And so medicine seemed like a good fit. And
Less well
along the
paid.
what's that?
Less well paid.
Less well paid, yes, but and anybody who watched the Masters this week would know that nerves are definitely an important part of the golfing. It was really terrific to watch. But along the way, I was very lucky to have great mentors. I think when I was a resident at Penn, I worked with Matt Stern and Howard Herdig, who are the movement disorder specialists here, and their approach to patients had a huge impact on me, and their just approach to people generally. It was, I think, I'm the chief of the movement disorders division here at Penn. I have effectively have the job that Matt established and inhabited for so long, and I think that like trying to be like Matt and Howard has always been something that's sort of like a guiding principle for me. They're wonderful people. And then I went and did fellowship at Rochester with Ira Scholson and Roger Curlin and Carl Kieberts in the Parkinson's study group. And I think watching Ira in action also was extremely important to me as I was coming up and seeing his way he organized large groups of people effectively was really impressive. His commitment to a few simple principles in terms of how to conduct clinical trials was something that really stuck with me. And so I think that even though I didn't have family, I had family in sport rather than in medicine. Uh, these people that I met along the way were had an incredible impact on me, and it sort of steered me towards the career I'm in now.
So it was so neurology was a sort of where you were gravitating to, then movement disorders by virtue of the people you met like Matt Stern and such like, and then
Yeah,
Parkinson's because that's where you did your fellowship at
yeah,
Rochester. Yeah.
yeah.
And
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 it has a lot to do with people. I mean, the content is interesting. I mean,
yeah.
who doesn't? I mean, everybody at some level is, perplexed and fascinated by the brain, but it was really these human connections that were drove it more than this sort of fascination with the subject matter. Both,
Yeah.
but the people were really important.
Yeah, and I mean, I completely agree with you. I think these sort of people who you meet at critical points in your life often direct you. So I met very early on the late David Marsden, who was very influential in my life, who was a great British neurologist who had a lot to contribute in the world of movement disorders. But it was the sort of interest and enthusiasm they had for the subject which took you in that direction, and the fact that they were very supportive of you, often at a point in your career when you weren't really very important at all. So, um,
Yeah, I mean, I remember Matt, well, Matt especially sort of like introducing me to people when he didn't, was he went out of his way to do it.
yeah,
And I was incredibly grateful and have become more grateful over time and, try to return pay it forward to our trainees now. It's uh
yeah.
it's an obligation, I think.
Yeah, so I think it's lovely to encourage people at all stages of their career and to treat everyone in the same way. So Andrew, when you went to Rochester and started working with the Parkinson's study group, how long had that been going at that stage?
So I guess the data top study was sort of started around 85, 87,
Right.
and the main papers came out, I think well, 93 was the main paper. And I
Okay.
was there in 96 to 98.
96 to 98. So the
So it
data
was,
tot study was the first big study they did, was it?
yeah. So
Yeah,
the Parkinson's Study Group, the infrastructure was really created to run the data top trial.
yeah.
And then it
And
was
just
such
remind
a,
us that that was a study where they looked at selegiline and vitamin E and placebo. Is
yeah,
that right? Yeah,
great study. It was a multi-center randomized controlled trials funded by the NIH,
right.
800 subjects, and it had they're testing two alpha tocopherol, which is vitamin E, and selegiline, which is a MAO-B inhibitor.
Yeah,
And I think that Langston's data had probably just come out, where there was a
yeah,
small pilot study suggesting an effect of selegiline on Parkinson progression. And uh, and Ira was leading this study, and he created the Parkinson Study Group infrastructure, including the partnership with the statistical team at the University of Rochester, drug delivery, what became the sites became the Parkinson Study Group. Um, and it was also a really novel intervention. I mean, there's a lot of interesting things about Datatop. The size of it at the time was really unusual for Parkinson's. It had a 2 by 2 factorial design. And this is sort of like clinical trial wonkiness, but a 2 by 2 factorial design effectively
so what does
tests.
that mean, Andrew, when you say 2 by 2? What does that?
Yeah, I was going to try to explain it best I can, and
Okay.
hope that it won't be too
Yeah.
bad. Um, they effectively run two intersecting clinical trials at the same time. You can look at the trial as being a trial of vitamin E versus placebo,
Yeah.
um, and you can also look at it as being a trial of selegiline versus placebo, because there were 400 people getting vitamin E and 400 people getting selegiline, and there are also 400 people getting placebo, one way or the other. But there are actually four intersecting groups where there was 200 who got placebo only, 200 that got alpha tocopherol only, 200 that got selegiline only, and 200 that got both.
Right.
So if you look at it from like top to bottom, it's placebo versus tocopherol. If you look at it from the side, then it's placebo versus selegiline. And you can analyze the arms as if it was just these trials of the single agents, or you can analyze it as it's like an interaction trial where there's the actually you think of it as having four arms, and the fourth arm is the combined approach. And there are statistical ways that you can sort of decompose it and recompose it if you want to. And so it's sort of the interesting design
Yeah.
I mean, their approach was to test the agents separately and leaving the option of testing the interaction as sort of like if the trial was positive separately, then you could look at the third arm at the interaction approach. So it was a novel design, also, conserve statistical power for doing, two things at once effectively. Um, and so a lot of interesting things about the data top in terms of logistics, in terms of statistical approach, and then the outcome measure, the time to needing levodopa
Yeah.
therapy was a new thing at the time also, and using survival analysis to test the effective drugs for Parkinson's disease, also a new thing. So lots
Yeah.
of innovation, infrastructure building and innovation.
And was that really the first? So prior to that, trials in the US, a bit like the UK, would have just been done on an individual basis. Someone comes up with an idea, or a drug company, they would set it up, run it, and then move on. This was the real first concerted effort to bring together a network of people to take drugs on sort of large scale trials into people with Parkinson's. That,
It was certainly the biggest and the most formalized and really a quantum leap in terms of a multi-center approach.
yeah,
There have been, I'm not an expert on this, but I'm quite certain there have been multi-center approaches for like cancer and cardiology previously,
yeah.
so it wasn't like a new
Yeah, no,
idea.
I
I
mean, I know
think,
in other specialties, so we're often accused of catching up in neurodegeneration with what
yeah,
other, especially cancer, have been
yeah,
doing for
yeah,
decades. But
yeah. But I mean, really audacious and like, you
yeah,
know, the ability to like think big and take risks, and really impressive.
yeah. And so the data dot study I remember as being very influential at the time because, as you say, Bill Langston had made this observation in these drug addicts in California that this toxin MPTP caused a form of Parkinsonism
Mm.
that worked through this monoamine oxidase inhibit and monoamine oxidase. So if you inhibited it, it slow it down in sporadic Parkinson's disease in the grounds that something like MPTP might be at the basis of sporadic disease. And the outcome from that trial was that it sort of slowed down, sledioline at least slowed down the time to take the
Yeah,
L-DOPA. But then the question was it disease modifying or symptomatic, i.e. making people a bit better?
yeah. And
Yeah,
um it was really it's so the whole like intellectual controversy that followed Datatop is also very interesting
yeah,
because the original paper showed unequivocally that people who were on selegiline but not on tocopherol.
yeah,
So there was no effect, no effect of null effect on of tocopherol on time to needing levodopa, being rescued for levodopa therapy. But unequivocally, the people who were receiving in the arms of the trial that received selegiline had a slower time to reaching the endpoint of needing levodopa therapy. So it was there was definitely the appearance that the progression of Parkinson's was being slowed down by
yeah,
selegiline. And there's also this complicated analysis that's in the 1993 paper looking at the effect of a washout, because there was already a question about whether selegiline might have a symptomatic effect, which was sort of tipping the scale and creating the appearance that there was a disease-modifying effect when it was really a symptomatic effect. And the
yeah,
washout was sort of showed some washout where there was some sort of tendency of the sledging patients to revert to closer to the placebo patients, but not that much, and
okay,
not enough to fully support the idea that this was a symptomatic effect. I think the interesting thing is that over time, the the idea that MAO inhibitors really exert their slowing effect on the time to needing levodopa through by a mild, modest effect on symptoms rather than by changing disease trajectory is really that has become the consensus view. But the evidence to support that is modest. And what
yeah,
we really know at the end of the day is that what we really learn from clinical trials is that at a certain dose, the drugs have a certain effect on a certain outcome measure. And
yeah,
mechanism is not well elucidated by clinical trials. It really comes from sort of corollary evidence from basic
yeah.
science and from other kinds of observational data.
Yeah. I mean, it's an interesting study because I sometimes give a talk about if I had Parkinson's, what would I do? And I probably would put myself on a mono oxidase inhibitor on the grounds that they're relatively innocuous. In the UK, they're cheap, and you can't quite get away from the fact that they might be doing something that's good to slow things down, even though the evidence isn't necessarily there. So
Yeah.
that was the beginning of the Parkinson. And then when you joined it, so there's a question here about in the Datatop study presumably all the tablets were the same. So the placebo group was, so everyone took a tablet or two tablets, and it could either be the selegiline, the vitamin E, or placebo. So they all looked the same. So the placebo group was the same for both the vitamin E and the selegiline
Yeah.
group. Yeah,
Double blind, double dummy, like you said.
yeah, yeah, yeah,
And so there's four tablets. You can't tell them apart. They're probably capsules, actually. I don't know for sure.
yeah, yeah,
Um,
yeah,
and so you wouldn't know you're taking everybody takes two capsules
yeah.
and you don't know what's inside of them.
Yeah. So it's quite interesting as we move to multi-arm, multi-stage trials that in fact Datatop wasn't that dissimilar in some ways in how it was constructed around looking at two compounds against a common placebo, if you like.
Yeah.
So Andrew, when you moved to start working with the Parkinson's study group, and as you say, the great sort of Ira Shulson, who did so much in this as well as in the world of Hunter's disease by setting these things up, what was your sort of remit when you arrived there? What was your project, or was your project more about thinking about trial design, or were you involved in a particular trial?
When I got to PSG in Rochester.
Yeah, yeah,
So it's like it was a big operation. There were must have been 20 people. It was in an old motel, a converted motel, down at the end of the campus, and there must have been, I don't know, 20 or 30 people working there at least. Uh, and Carl Keeperts was definitely there, very active. Bob Holloway, who's the chair of neurology at Rochester, was sort of peripherally involved. And there was an army of coordinators, and I was assigned to work on the Teva trials, the resagiline trials,
Yeah.
and it became Tempo and Presto. Um, and I was the assigned to be the medical monitor for the Tempo trial, I guess Tempo and Presto.
Okay.
And so I learned a lot about safety monitoring at that point. Um, Ira introduced me to the folks at Teva, who are the sponsors, and I was fresh out of residency, had never done any clinical trial work at all. And then they asked me a few questions about like what I thought about like the FDA regulations, and I didn't know what the answers were at all. And they were like, "Are you sure this guy's going to be okay?" And they made me take a course, like a training course on like medical safety monitoring and clinical trials, which was actually a really good idea. And I was glad I did it and learned a lot on the job, um,
Yeah.
and actually done
So
a
is
lot
that
of
how
safety
it
monitoring
worked?
work since then.
So the Parkinson study group worked by people coming to them with drugs that they would like to test. So Teva, a big pharmaceutical company, would come and say, "We think Rosagiline, a monoamine oxidase inhibitor, we think it might have disease-modifying therapies, or we want to test its efficacy. Can you help us set up a trial to do that?" So it wasn't that, Andrew would turn up saying, "I've actually got an idea for a trial. Can you help me run this trial?" It was more pharma that came to them, or how were drugs selected and then trialed?
Yeah, I think, I mean, a lot of it was, there was this Parkinson's study group right from the beginning. Again, something that, was Ira's vision, had a lot of structure. It
Yeah,
had bylaws and it had an executive committee. And no doubt, and Stan Fon
yeah,
uh, also was an, early leader in the Parkinson's study group. And so, and Ira and Stan were the chair and co-chair of the executive committee. And the executive committee consisted of people, like you and me, basically, who were
right.
supporting their efforts. And um, so the trials, I mean, certainly Ira had a lot of input into what was picked and what wasn't, but eventually, it was sort of a democratic process where the trials would come to the EC and they would decide whether there was a good trial for the Parkinson's study group to take on or not. I think, one of the things that happened was in the wake of the success of Datatop, the company that made Selegiline sponsored some follow-on trials, and that was a nucleus of business for the Parkinson's study group and supported the infrastructure. But then, like Cliff Schultz brought the vitamin the coenzyme Q trials to
Yeah,
um, the PSG, and there was a really influential clozapine trial that Joe Friedman ran. So
right.
the I'd say the portfolio was a mixture, but there was there was always an industry presence, and a public-private partnership was always part of the PSG.
Okay. But it wasn't part of the Parkinson study group to sort of it's I mean, I'm part of this linked clinical trials run by Cure Parkinson's where they go through a whole list of drugs and select each year drugs which they think should be going to trial. So there was no formal process where every year you would have a meeting and run through a portfolio of drugs or agents which you thought might be useful to
Yeah.
go to trial.
No,
It
there
was more
wasn't like a rational
proactive.
selection process like that. And it's a good idea
Yeah,
for the NetPD effort in the United States. It must be something equivalent in Britain. There was
yeah.
something called Synapse, which is a selection process to identify drugs that would go forward into the trial pipeline. But
Okay.
to my knowledge, I mean, I was very young at the time and I might have missed it.
Yeah.
But I don't think there was a formal selection process. I think it was more opportunistic.
Okay. And you were also, as you were clearly saying, doing drugs which were used for symptomatic control or for aspects of the disease, not all disease modifying. It was therapeutics for Parkinson's in the more general sense.
Yeah.
And on the back of that time you spent there, Andrew, what did you do next? So how did that inform you as to what you wanted to do and where you wanted to take your work in Parkinson's?
Yeah. So after I was a fellow for 2 years,
Yeah.
and yeah, it's an interesting thing. I think our fellows sometimes stay for 1 year, sometimes two, and sometimes three. And I sometimes look back and say two was good, but maybe three would have been better. I don't know what you think. I think that when I see people who are really successful in their mid-careers, I think sometimes they've invested more in their early careers to really get a solid foundation. But I did 2 years. and came to work at Penn. I got came back to Penn where I'd been a resident, and with the idea that I would work sort of do clinical trial work along with Matt and Howard, maybe do more in-house trials, maybe do a little more biomarker work, I suppose, and also be a busy clinician. I see patients now like 4 half days a week. I had clinic this morning. I just finished, and um, and I saw a patient who was originally a participant in QE2, had Parkinson's for my patient for 20 years. I just
Yeah.
left the room with him. He's not good, he's good.
Yeah.
Um, and he's on selegiline,
Yeah.
which is a funny thing. Actually, I've had people on selegiline for so
Yeah.
long. And so And then I got a K-23 grant from the AHRQ, which is an organization that sponsors health policy, which is actually, I think, in the process of maybe shrinking a bit right now, but it was an important agency for understanding the effects of health policy on health in the United
Right.
States. I think while I was at Rochester, I worked a lot with Ira and Carl for sure, but I also worked a little bit with Bob Holloway, who was interested in health economics and health policy. And his mentor was a guy called Al Mushlin, uh, who happened to be a residency classmate with Howard Hurdig just by chance,
Right.
um, who was an expert on health economics. And so became interested in quality of life outcomes.
Okay.
And I got a career development grant from the HRQ to study patient-oriented outcomes, PORs, in
Okay.
that are especially related to cost effectiveness analysis
Okay.
in Parkinson's. So the early part of my career, I spent kind of doing health economics related to Parkinson's.
Okay.
And it was
Well, that's
interesting,
very
but
much
it felt
ahead
like
of
it
its
didn't have a lot of
time.
traction. I moved back towards clinical trials later on.
Yeah. But I mean, that's quite ahead of your time because I mean, nowadays everyone talks about patient-related outcomes. Well, as most things in medicine, people always talk about things that they've been newly been discovered when they have been around for some time. But patient-related outcomes have become quite a very important aspect. In some of our trials, which I talked to the FDA, are very keen on this. So it's interesting how you were involved with that early on. And I can see how your career is sort of slowly meshing together. You have this interest in drug trial design. How do we do trials? And then linked to that is clearly what outcomes. And that is still an area which is obviously still unresolved as to what are the best outcomes for some of these trials. And obviously it depends what stage they're at. But also, especially my own area of cell and genes, health economics becomes a really big thing because, you can prove whatever you like, but if the cost of the drug is so high, no one will ever have it. And so those aspects as well. So during this time at Penn, you were sort of slowly forging a path of health economics, drug trials, and also thinking about outcome measures and biomarkers you were talking about. So I mean, this is something which I think you've got more and more involved with as the years have gone on. I may be
Yeah.
wrong, but I mean, I think it'd be interesting to be here back in those days. So at the sort of turn of the century makes us feel very old. But I mean, biomarkers were pretty crude in those days compared to what we do now. And so, in your experience, what has at that stage in your life, what were the big challenges with the biomarkers, and how have you seen that evolved over the years?
Yeah, so I think we sort of undersell our biomarkers a little bit because we've had dopamine imaging for
Yeah,
a long time, and it's actually a really good biomarker for diagnosis for sure.
yeah,
For prognosis, not prognosis, but following disease state,
yeah,
it's very good for that too. I think it's never been very good for testing drug effects in terms of slowing disease progression. But maybe you blame that on the drugs rather than the biomarker. If we had a drug that really slowed down the progression, maybe the death scan would look a little better.
yeah,
And then there are other, there's related to DAT scan. There's the current one we have ioflupane, which is an iodine-based SPECT tracer. There are PET tracers which have better spatial resolution. There have sort of come and gone SPECT tracers that have other features that make them desirable. Either they maybe like use technetium instead of iodine so that they're a little easier to manufacture and cheaper. But DAT scan has been the one that's had the most legs so
yeah,
far. Um, and I think that it definitely has usefulness in terms of diagnosing Parkinson's versus certain mimics, but not all, obviously, um, and potentially has some role in early detection.
yeah,
And I guess that's the next thing that is sort of I guess I had been interested in smell testing. I
Right.
wasn't I sort of fell into it because Matt Stern had been interested in smell testing. There was this fellow named Dick Doty at Penn who developed the University of Pennsylvania smell identification test,
Yeah.
and he and Matt had tested it in a bunch of Parkinson patients and found that I guess this has always been known, but that about 85 or 90% of the Parkinson patients had a profound smell defect.
Right.
And it happened earlier. I mean, it was really present at the time of diagnosis, and presumably it evolved prior to the time that patients were diagnosed with Parkinson's. Uh, but nobody knew quite how long or how, what the time course of that was. So
So just on that, I mean, how did that, I mean, was that smell test set up because this was thought to be something that needed to be put into a sort of quantifiable test? Or was it just that, I mean, how did people get interested in smell? How did Matt get interested in it? And then how did you get interested in it? Was it a sort of concerted program of work, or was it merely sort of interesting to look at and then suddenly discovered it was actually proving to be very successful?
yeah, I'm sort of trying to. It was a long time ago. I'm trying to put the pieces back together, but the way I remember it is that Dick Doty had been interested in smell, sort of not really related as it relates to Parkinson's.
Yeah.
He'd been interested in it in like factory workers, uh, or in smokers, and had developed this sort of scratch and sniff smell test, which was easy to use and could be deployed to test populations.
Mm.
Um, and I guess he must have had a conversation with Matt at some point where they said, Matt said, my patients all can't smell. We should test it. And then they did test it in a few like, seminal papers that were probably came out in like the 80s, I think. I have to go back and look, showing that there was this really profound smell deficit in even in the earliest stages of Parkinson's.
Yeah.
And then Matt brought this up with me, and since I was interested in cost effectiveness analysis and sort of diagnostic strategies, I had for my, I guess it was for my thesis or work related to the master's program that I took, I created sort of a back of the envelope cost effectiveness analysis of smell testing as an adjunct for diagnosing Parkinson's disease and how it might add to the effectiveness of diagnosing. And I presented to my mentors who were not neurologists as part of like the program I was in, and they were like, "Wow, this works incredibly well. This works so well. We really can't really believe it. Actually, I think it's too good to be true."
Yeah.
And I went back to Matt and said, they thought this was like almost like too good to be true.
Yeah.
And then Matt said, well, I bet we could use it to like find people who don't have Parkinson's at all, but are at risk.
Yeah.
And this could be part of like picking people who have prodromal or premotor Parkinson's out of the general population and and um so identify people at risk who could then sort of be followed to see if they develop Parkinson's. And this was sort of just a casual comment that Matt Stern made sort of standing around the nurses station one day. And then I guess Matt must have said the same thing to Ken Merrick at some point. And then there's a study we that really, Ken sort of took this idea and sort of, said we can implement this on a big scale. And so he launched this thing called the PARS study, the Parkinson at Risk study, where we sent scratch and sniff smell tests to like just mailing lists, firemen, nurses, and there were some first degree relatives or our patients in our clinic. There are 10,000 of them. We sent out 10,000 smell tests. We must have like bought Dick Doty a new car because he had a company that made the smell tests.
Yeah.
And but then sort of I think the rest is sort of well known that the PAR study identified these people that had a bad sense of smell. They went up to New Haven where Ken and Dana Jennings and John Seibel were doing uh, SPECT imaging. It wasn't fluke painting, it was a different um, SPECT tracer. And so we did sort of this combination of smell testing and DAT imaging, and identified an at-risk cohort. I guess it's also around the same time a group in the Netherlands led by Ponson had done the same thing on a small scale, and we sort of stole their idea and made it much bigger.
So
Um,
this was really the first. So I mean, you've already talked about the first concerted attempt to sort of do proper big studies in Parkinson's, but this was the first major inroads into this concept of pre-motor Parkinson's, prodromal Parkinson's disease, pre-motor, whatever you'd like to call it. So this was the first real study that
In
brought this concept to the sort of table on a large scale.
I'd say, I mean, I think I would be remiss if the Ponson group had done to publish this in Annals of Neurology. Maybe we'd known about it for sure
Yeah,
before we launched our study. And Schenk had definitely published on RBD.
yeah,
And so it wasn't like we were, you know, doing this completely independently, but it was bigger. It was
yeah,
bigger and multi-center, and it didn't rely on REM sleep behavior. It was all sort of centered on the idea of olfactory deficit. And
yeah,
I think we were really lucky because it turned out the olfactory deficit turned out to be extremely predictive. Um, and it was really an early deficit. It really does seem to precede the DAT deficit in a lot of cases.
okay.
And just by chance, more recently, we've learned that it's incredibly well connected to synuclein pathology.
Yeah.
Uh,
Yeah, well, I was going to come
so-
on and discuss that because I mean, one of the great, one of the great challenges has always been that when you have these tests, you can sort of say now we can pick up people with Parkinson's before they've got Parkinson's, and then one of the questions I often get asked is, well, would you go and screen the population for it? And of course, one of the problems is you could screen the population. Obviously, if you've got a good therapy, you could use it in that population. But if you don't, what do you do with that information? So, you get screened for colon cancer and breast cancer because you can do something about it. But then it's always difficult because I think, well, if you don't do it, then you'll never be in a position to be able to implement a disease modifying therapy at the earliest possible time. But also, I can see the dilemma that if someone came to my house and I did all these tests and they said, well, Roger, you've got a very high chance of developing Parkinson's in the next 10 years, what do I do with that piece of information given there's nothing you can do about it? So I suppose in these early studies, it was more about exploring whether you could find a precursor state, if you like, to Parkinson's. And I guess the ethical aspects of it were perhaps less controversial because you were really trying to just define a state rather than say anything more about it, and it was still experimental.
yeah, so I think that things have progressed a lot since I started doing this in terms of the ethics for sure. Um, we didn't tell anybody any of their results,
Okay.
and we enrolled a certain number of like decoy subjects as specified by our IRB so that people couldn't figure out what group they were in or what their results were based on sort of whether they're asked to come back for follow-up or not. So
Okay.
uh, and this was the same strategy that other people were taking at the time
Okay.
it was, really different times. I think that the idea that patients should be able to get their research results returned to them and know what the results are was something that really wasn't, I think, thought to be the ethical thing to do. I think the ethical thing to do was that there's so much uncertainty around what these tests mean. They don't have a clinical utility, as you were saying, and so the
Yeah.
patients, it creates distress for the patients in order to, if they get the information, so that we shouldn't do that. So we had barriers set up so the patients wouldn't get the results, and the study was designed so that they couldn't infer what group they were in by sort of what procedures they got.
Okay.
It's amazing how things have changed.
Yeah, 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 yeah. And of course now with electronic, well, I suppose it's research. I was going to say with electronic health records, it's think people can have much more access to information. So at this point, Andrew, we're sort of moving through. You've got this expertise in clinical trials. You've got expertise in health economics. You're thinking about quality of life. You've started to develop biomarkers around, well, imaging, which was already there. This, hyperosmia, the sort of smell test. Uh, and then how did this evolve next? So I can't. So you're heavily involved with the PPMI, and I'm not quite sure when that came into existence and how that relates to what we've discussed now. So I don't want to jump ahead too far, but
Yeah.
based on all of this work, what was the next sort of major step in your career in terms of research into Parkinson's?
Well, so I'll get there in just a second. So PPMI, this was all happening. Um, PARS started probably in 2007 or 8 or so, and
Yeah,
PPMI started in 2010.
yeah,
And I guess I knew Ken from PARS, and so
yeah,
he launched PPMI, and I was lucky enough to come along with him and Dana Jennings, and a lot of the team is still intact actually.
yeah,
If you look back at the old pictures, you see a lot of the same faces that you see now when you go to the PPMI annual meeting. Uh,
yeah,
but the thing that I did was I left Penn and went to work for Eli Lilly.
Okay.
I mean, I don't know if you know, but I around 2012, so
So why
right
did you
around
do
the
that,
time that
Andrew? That seems like quite a major change, given you seem to love patients, and of course, I suppose Eli Lilly may have allowed you to carry on doing clinics, but
yeah,
what provoked you to do that? And
yeah. So there is Eli Lilly, is a great company, and
yeah,
they had recently um, they there was recently purchased a small company, biotech startup called Avid Radiopharmaceuticals in Philadelphia. And Avid Radiopharmaceuticals was the brainchild of this incredibly talented guy called Daniel Skravanski, who's now the chief scientific officer at Eli Lilly, uh, who had the idea that you could take basically the same technology as the Pittsburgh compound B that images amyloid
yeah,
um, pathology in Parkinson's, I
in
mean, in
Alzheimer's,
Alzheimer's disease.
yeah,
And you could just change this a little bit so that it's a fluorinated tracer rather than a carbon 11 tracer. And it could be transportable and scalable and commercializable. And you could effectively detect amyloid pathology in people who had either dementia or else or MCI and make a
yeah,
more accurate diagnosis of Alzheimer's disease based on pathology. And then all part of this sort of long-term strategy that you could potentially remove the amyloid pathology, treat the Alzheimer's disease, demonstrate this on the PET biomarker, and it would also have a clinical benefit correlate. And of course, this is sort of lecanemab and donanemab.
yeah,
What
yeah.
they finally, this sort of strategy, I mean, I guess some people say that these drugs don't have a dramatic effect, but I think it's amazing that we have therapy that gets at the pathology of neurodegeneration at all and alters the clinical course. And so this was sort of this Avid Radiopharmaceuticals was sort of the start of this, at least part of it. Uh, and it's in Philadelphia. It was right across the street from Penn, actually. It was because Dr. Vronsky was a postdoc in in the Trojanowski Lee lab
Right.
when he started the company. And I guess one day somebody from the company came in to my office and said, "We're looking for a somebody to do a medical director role, and we're going to start working on Parkinson's as well as Alzheimer's. Do you know anybody who might be interested in doing this?" And I said, "Well, I might be interested in doing this." They had this
There's one
paper in
person who's
JAMA.
very well qualified for that, and that's
Yeah.
me.
And it was biomarker work. It was really at the cutting edge technology, a really great group of people.
Yeah.
And I continued to see patients at the VA actually during the
Okay.
time I was at Avid. And it was wonderful. It was a great experience. 5 years are really well spent. I'm really glad I did it.
And so that work was really around developing imaging tracers
Yeah.
and that was that because that was not just amyloid, that was for tau, for synuclein and the protein in Parkinson's, or was it more about developing better markers for amyloid?
Yeah, Avid sort of fit into the Lilly um, sort of universe
Yeah,
in a few different ways. They definitely had a tracer discovery programs,
right.
um, better amyloid tracers, so next generation amyloid tracers that didn't have, say, for example, the off-target binding in the white matter that's always been sort of created some challenges for reading the amyloid scans.
Yeah,
They had just bought fluortaucipir, the technology for what became fluortaucipir, the first tau tracer for
yeah,
NFT pathology in Alzheimer's disease, and potentially for other like 4R tauopathies, but it really works much better for
yeah,
the neurofibrillary tangle 3R,
yeah,
4R tauopathy. Um, and then they were developing PET ligands for the dopamine system, especially um, the, the dopamine system, sort of analogous to DASCAM, but not quite exactly the same.
okay, okay.
Um,
But it
and
wasn't the protein, it was the dopamine system they were
the
trying to.
doping system. And
Yeah,
then they
yeah,
also, it just goes to show how hard a problem this has been, but they had a nascent synuclein imaging program also, which and they're trying to develop synuclein tracers, but synuclein imaging, developing good molecular imaging technology for this just turned out to be super challenging. And
yeah,
I
yeah,
think I think there's some progress that we saw at ADPD couple weeks ago, but even so, it's not quite there yet. It's getting
Yeah.
there, but not quite there yet.
Yeah.
And
So
then
what did
the
that?
other thing they did was they did image analysis for all the clinical trials.
Okay. And were you in charge of those clinical trials or?
You know, every place I've been, I've always been part of a team. I like to
Okay.
you sort of talking about it as if it's like, you know, my work, but it's really not. I'm just, fit into these larger
Yeah.
teams. And I think actually seeing really well functioning teams at Lilly was a
Yeah.
really
Yeah.
good learning experience for me because they had scientists and clinicians like me, and they paired them with managers who were professional managers who like were really good at like timelines and budgets and
Yeah.
so the level of professionalism that went into like making sure the ex project was executed in an
Yeah.
efficient way was paired with the scientific expertise, and I think that was really good lesson about how to get things done. You
Yeah.
don't leave it to the scientists because they might you know dither, and but they was not happening. But they had professional managers. Um, the so that was really a good experience. And there's also like a focus that comes with it working in industry where you really need to, at the end of the day, you need to have a product that works. And
Yeah.
so, you sort of there are scientific sort of side trails that you might get diverted into, but at the end of the day, you've really got to stick to the main trail. And that was good experience too.
So it was a
Um,
very formative time this time in Eli Lilly because you could sort see how to run trials very efficiently with these teams with different expertise, and you could also see that they had a very clear end game. What we wanted
yeah,
to deliver, which I
yeah,
think, as you say, sometimes in academia it's sort of slightly, people clearly see where they're going, but they're not quite sure what the end game looks like, and then they slightly move the goalposts and such like.
yeah.
So at the end of that 5 years, Andrew, you then came back to Penn, did you?
Yeah.
You
I would
came
just like
back
to say
across the
that
road.
they say that like we in America, I don't know if you say it in England, we say like that the pharma is the dark side. Like that's like,
Yeah,
somehow like morally, suspect. But that's so not true about my colleagues
yeah.
that I met at Eli Lilly. They're some of the most honest and ethical people I've ever met. And I think that's important.
Yeah, and I think I grew up with that view that pharma was, if you like, the dark side. But ultimately, if any therapy is ever going to make it to clinic, you have to work. You're going to have to work with pharma because they have the money and the wherewithal to actually do this. So you have to partner with them, and I think that's a very useful way in making sure that therapies can make it all the way there. So I mean, I think it's great that you've got experience from both sides. And when you came back, I mean, some of the questions which are, emerging in our chat are around, this later phase when you've come back to do clinical trials, and obviously with these new ways of testing for Parkinson's. Um, I suppose one of the questions I would have is when you came back to do more clinical work, how much had the landscape changed during that period of time? Because you said back at the turn of the century, it was much less patient centric. You were sort of, if you like, bringing people into trials. There wasn't that much feedback. But how had things changed now so that patients were much more at the centre of the trials? And what was the sort of changes you'd noticed in trials or were beginning to happen in trials, which perhaps weren't there before you left to go to Eli Lilly?
Yeah. So I think if you sort of take the idea of like disclosure of research results,
Yeah,
that just as an example, I don't know if you how you feel about this, but when I got started, I think guided a lot by examples from genetics, there was a strong reluctance to share the results with the patients that the samples came from, and that there was a feeling that it wasn't useful information and it could be frightening um and that I think way of thinking has really done a complete change. I think that there's a strong recognition that the patients have donated these samples and sort of they don't own them. Actually, they really belong to the researchers, but they do deserve to get the information back about their research results. And I think that this has become more the norm than the exception. So it's really been a complete change in this. I actually, and there have been several studies looking at, say, ApoE status in Alzheimer's disease, which show that actually patients can understand these results quite well and behave quite rationally when faced with either you know a positive or a negative result. Um, my feeling actually is that, and this is going to be a little bit controversial, I suppose, is that I think we need to sort of even look a little farther, because I think there are definitely times when it's in nobody's interest for the research results to become known right away. And you're going to learn a lot more, at least temporarily, by shielding both the investigators and the participants from knowing what their research results are. I mean, one really simple example is if you know that you have a positive DAT scan and you're a prodromal individual, that you're likely to sort of interpret that little twitch in your hand as being meaningful. Whereas if you knew your desk didn't know your desk was positive, you might think nothing of it. And you might check some more boxes on a PR patient reported outcome than you would have otherwise. And I know as a doctor rating patients that when I know that somebody has, positive scans or positive SAA for now, if, for example, biomarker evidence that there's something going on, that I might give them a point or two more on a rating scale that I would have probably withheld in the past. And so I think there are times when patients and physicians both need to sort of own the greater good and not get the results, at least temporarily. I think that we've swung so far in terms of giving everybody results in real time that we potentially are missing opportunities to generate knowledge that's going to be really valuable in the future. So, but I think a lot of people don't feel this way. I think a lot of people feel like we should just to give the results and the information will take care of itself. But I think that this is still an evolving concept, and
Yeah.
personally, I think it would be nice if we landed somewhere a little more in the middle than
Yeah.
where we are now. But it's definitely been a great change from where we were, in a positive change from where we were 10 years ago.
Yeah. So, I mean, I guess, as you say, one of the big changes has been that patients are much more informed about things as they're going through work, through trials and research in a way where one was a bit paternalistic and protective in the past in a way which perhaps wasn't that helpful. So I want to sort of bring it onto this sort of, so more recently, obviously we've now moved to a stage. So, if I just sort of summarize where we've got to with this, Andrea, I mean, you've obviously worked on trial design, you've worked on biomarkers, prodromal Parkinson's disease, you've seen it from the pharma side of things. You're now got patients much more at the center of things, sharing information with them. And more recently, because we've got a limited amount of time, we've got this new seed assay. So this is this alpha-synuclein seed assay, which is a way of diagnosing people who carry abnormalities in alpha-synuclein, Parkinson's, and such like. So this has obviously been an exciting discovery. It's generated a lot of discussion. So I thought it might be useful in this last sort of part of our discussion to say, how much do you think that's changed things and will change how we view Parkinson's and trials going forward? Do you think it will dramatically change how we do things, or do you think it's just another useful tool which we'll have to integrate into everything else that we've got?
Yeah, I think it's a brand new thing, and I think we're learning.
Yeah,
Um, it's been known that Lewy pathology causes Parkinson's disease since around 1915. And
yeah,
that was sort of Frederick Lewy's paper was like 1912, I think, a
yeah,
really, really long time ago. It's been known that synuclein is the main constituent of Lewy bodies since around 1987, I think. Um, I don't remember correctly, but it's
yeah.
been a long
97. Well,
time.
97 was when the families were described. 98 is
Yeah,
when
that
they
was
discovered it.
97. So
They put it.
2004, I think. You're right. It was
Yeah,
a little, but
yeah,
still a long
yeah,
time.
yeah,
Um, the Spallanzini paper.
yeah,
And so it's been a long time. So this is not, but what's new is that we can see it in life,
yeah,
in living patients. And it's led to a reconsideration of what Parkinson is, really, but really along the same lines as what Frederick Lewy described 100 years ago, that it's a not just a clinical syndrome, but principally a biological syndrome, a brain degeneration that produces characteristic clinical features. And so this led to these there's sort of a recognition from several different quarters of a biological way of defining Parkinson's disease biologically based on synuclein presence of synuclein pathology and neurodegeneration. And obviously, there's a couple different competing schema, but they're really similar in those core features. So I think that the idea that there's a emphasis on the a biological definition of Parkinson's is actually a area of consensus
So just so we got
among
this clear, Andrew. So biological would be so clinical would be I've got a tremor, stiffness, slowness. I've clearly got features that's Parkinson's.
yeah.
Biological would be I don't really care what you look like. I just know that if I take your fluid and I do this assay, you're positive on the seed assay. You have an alpha-synuclein pathology. So biologically, that is the condition you have. What you look like is obviously can be helpful, but it's not defining what I'm calling this disease.
I would say yes and no. I
Okay.
think that's sort of like a fringe case. That's sort of, I think that the case that I'd like to focus on is that you've got a tremor and rigidity and bradykinesia, and that you also have the key pathology, the synuclein, underlying synuclein pathology. That's really that's the way we've always thought about Parkinson's disease. It's a
Yeah.
clinical pathological entity with these typical core features and underlying synuclein pathology, Lewy pathology, and this is nothing new. I think
Okay.
the idea that it extends to people that have a certain biology but have no clinical features at all, that's sort of a bit of an edge case, but a case that people are really focusing on a lot these days. Um, and um, and then the idea that you have uh, clinical features, but no synuclein pathology, like some people do that have like a LRRK2 variants, for example.
Yeah.
I think that's also sort of a fringe case and represents a small number of people. I think these are challenges to be worked out in terms of the
Yeah.
biological classifications, but I think that the core of it, that Parkinson's is a clinical pathological entity with tremor, rigidity, bradykinesia, and underlying synuclein pathology, that's sort of that's nothing new. I do think that understanding, especially what these asymptomatic individuals mean and how to think about them, whether they have something that's inevitably going to lead to a clinical syndrome that's going to produce disability or not. That's something I think that's an open question, one we're going to answer in the next few years with studies like PPMI.
Yeah. And I suppose for our sort of wider audience, I mean I would guess at the moment we could summarize it by saying that these seed assays are very exciting. They're more in the domain of the research area than sort of clinical practice. But they may well become more, in the way we use DAT scanning to help us make a diagnosis of Parkinson's, these may well become more routine as time goes on.
Yeah,
Um,
I think it's like you said earlier that in the setting of effective disease modifying therapies, the 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 need, the use case completely changes.
yeah.
But
Okay.
in the absence of that, it's knowledge gathering and research at this point.
Okay.
But
So
important
just to
important
sort of finish
research, the
up,
research that I think that I'd hope people would think is valuable enough that they'd want to participate in.
yeah,
People
I
that
know it gets
are
people
interested.
quite worked up that this idea that we talk about alpha-synuclein neuronal disease or right, and we get rid of the term Parkinson's. But I think you and I are probably on the same page with this, that it's too early to make these changes. But the fact we have a biological marker is very helpful. So in the last couple of minutes, Andrew, I wanted to say, how is this going to help us? So obviously we could treat people earlier in the condition with more confidence in the sense that they have this positive seed assay in the context of the possibly right clinical phenotype. But I suppose, one of the questions has been that alpha-synuclein has been the target for many therapeutic interventions so far in Parkinson's, but none of them have really delivered so far. So is it because alpha-synuclein isn't actually at the center of Parkinson's and we've got that slightly wrong, or our therapeutics have yet to work out how to best deliver to the brain to get rid of the protein that's driving the condition?
So we have 3 minutes left. I'm going to take a quick stab at this. Uh, Personally, I think the synuclein is important for Parkinson's. I think you see that from the families that have the autosomal dominant Parkinson's, and from the families that have gene duplications and triplications, and then from the basic science research that came from groups like John Trojanowski and Virginia Lee and others. Uh, I think there's strong evidence that synuclein is important for Parkinson's. And then I just think we haven't, it's a good target. I just think we haven't hit the target as
Yeah.
squarely as we could. I think there's a hint from the recent Roche study that maybe there's something going on there. I
What the
would look at,
long-term follow-up?
Yeah. And I was sort of,
Yeah.
in the Alzheimer's experience where they took swing after swing at amyloid until they finally hit it, I think is encouraging. I just was going to end with the experience with immune modulating therapy in multiple sclerosis. I was
Mm.
recently had to look at this a little bit for something. And the original like beta seron that was original like treatment for multiple sclerosis worked just a little tiny bit
Yeah.
and had lots of side effects. And over a process of about 15 or 20 years, these immune modulating therapies iteratively got better and better to the point that now we have drugs that are really effective. And I think that so I think the idea that we're going to hit a home run on the first swing for anti-alpha-synuclein therapy is probably not borne out by these examples either. We're beginning to see this a little in Alzheimer's, but we've certainly seen it to maturity in MS, where you get sort of the idea that you're on the right track and you open the door, and then you sort of iteratively get better at hitting the target until you have really clinically meaningful drugs for patients. And I think that's what the future for Parkinson's synuclein therapy looks like.
Yeah, I mean, I think that's a great way to end because certainly in my clinical career, the treatment of MS in the late 1980s used to be steroids as a sort of just speed up recovery. And then they started using immune therapy, just as you said, and it didn't work. And then they realized they were going too late and they were going in the wrong type of patients. And now, as you say, we've got a whole raft of therapies that work very effectively. But you learned as you went along. And I suppose that's one of the great things we've learned this afternoon, Andrew, that you've learned a lot as you've gone along through your career and how things have evolved and how we understand not only how we should do trials, how we should integrate all of the various tools that we have, biomarkers with clinical, with imaging, the value of a team, which I think is so important in a world where currently individuals seem to be the most important aspect of it rather than the team that delivers and all has expertise. So I want to thank you very much for that. You've raised many very interesting topics about information that should be shared with patients and with clinicians and who should have access to what and what that information could be used for. So that's generated quite a lot of discussion about who should know what. But I want to thank you very much for, fabulous conversation this afternoon, an incredible.
Thank you. And thanks to everybody at the WPC for making this possible.
Yeah. So it's been absolutely terrific. So thank you very much. I'd like to thank again Supernus Pharmaceuticals for funding this. Without it, wouldn't be possible. I just want to highlight that we have another one of these research spotlights in June, same time, which will be 4 p.m. my time and 11 a.m. Eastern American time, and that will be Ashley Harms from the University of Alabama, who will be speaking about T cells, so the role of the immune system in POGSI. So we actually ended very well, Andrew and ourselves, by talking about the immune system and T cells, which are very important in MS. What is their role in POGSIs? So thank you all very much for joining us this afternoon. I want to thank Andrew again. I want to remind you all about Phoenix 2026. That's when the next WPC Congress will be. That will be the seventh one, May the 24th to 27th. I was in Phoenix last month. It's a terrific place to go. So come and join us then. And thank you all very much for participating this afternoon, but especially Andrew for a fabulous 1 hour conversation. So thank you all very much.